All right, let's dive into a professional's guide to urine drug screening. If you're a medical lab student or you're heading into that career, think of this as your masterclass. We're going to break down the entire process, from that very first moment of collection all the way to the final report that can and does change lives. So let's get right into it. Welcome to the Bioscan Hub channel. If you find this guide helpful, and I'm pretty sure you will, make sure you subscribe to support us. That way, you won't miss any of our future explainers built just for medical and lab students like you. So here's our game plan. We've got 4 key parts. First up, we're going to tackle the big question: why urine? Why is it the go-to sample? Then we'll get into what I think is the most important procedure of all, the chain of custody. After that, we'll talk about how you, the professional, can spot a tampered sample. And finally, we'll wrap up by looking at the huge responsibility that comes with analyzing and reporting the results. Okay, part 1, why do we use urine? Before we even get into the how of doing the test, we really have to understand the why behind it. So why is urine the gold standard for drug screening? Well, sure, it's easy to collect and it's non-invasive, which is great. But from our perspective in the lab, it's the next 2 points that are absolutely crucial. First, drug concentrations are way higher in urine than they are in blood. This makes our job of detecting them much, much easier. And second, there's the detection window. Drugs and their metabolites can hang around in urine for days, sometimes even weeks. This gives us a much bigger picture of a person's recent usage history, not just what was in their system an hour ago. But, and this is a big but that every student needs to burn into their memory, there is one major exception: ethyl alcohol. Now, you can find alcohol in urine, sure, but blood is always the preferred sample. So why is that? Well, it's because blood alcohol concentration is a direct, reliable measure of someone's current level of intoxication, and that's usually the critical question, right? Especially when legal issues are involved. A urine test just tells you if they drank recently, not how impaired they are right now. And that brings us to part 2, the absolute procedural heart of everything we're talking about today, the chain of custody. Let me be blunt. If you get this wrong, even the most perfect lab result is legally worthless. It's that important.
So what is it exactly? A chain of custody is basically a legal document. It's an unbroken paper trail that proves who had the sample and when. Every single person who touches that sample, from the collector to the lab tech, has to sign and date it. There can be 0 gaps, no questions asked. This is how we guarantee that the sample we're testing is the exact same one the donor provided, with absolutely no chance of tampering.
Now, as a lab professional, your role starts right here. You're not just a collector. You are the first guardian of that sample's integrity. First, you confirm identity with a photo ID, no exceptions. You then ask the donor to remove any outer clothing, like jackets, to make sure nothing is being hidden. Next, you secure the collection area itself. This means adding a bluing agent to the toilet water and putting tamper-proof tape on the faucets. Why? To make it impossible to dilute the sample with water. And after collection, that toilet is not to be flushed. Every single step is about maintaining absolute control. The process keeps going with the donor right there with you. You're the witness as the urine is transferred to the lab bottle, and you're inspecting it the whole time. Then comes the first huge scientific check: temperature. You have a 4-minute window, and that's it. That sample absolutely must read between 90 and 98 degrees Fahrenheit. If it's cold, that is an immediate giant red flag that the sample probably wasn't provided fresh, right there on site. Okay, finally, we seal the deal. And I mean that literally. You're going to put a piece of tamper-evident tape over the cap, and, this is so important, you have the donor initial that tape. It's their proof that they saw you seal their unaltered sample. Then, you both sign the form. Every signature, every piece of tape, it's just another unbreakable link in that chain, making sure that sample is secure all the way to the lab. All right, so you've nailed the chain of custody. Let's move on to part 3, ensuring sample integrity. This, my friends, is what separates a good technician from a true professional. It's the ability to scientifically prove a sample is invalid, even when the procedure was followed perfectly. So this raises the big question, right? Adulteration, substitution, dilution. With all the ways people try to cheat, how can you be sure the sample you're looking at is actually valid? Well, it's our job to have the scientific tools to catch them. And here's just a short list of the things people will try. We're talking common household stuff like soap and bleach, all the way to more sophisticated chemicals like oxidants designed to destroy the drug metabolites. Or they might just try the old-fashioned way, drinking tons of water to dilute their sample.
The good news is our lab tests are specifically designed to catch these kinds of things.
In this table right here, this is your clinical checklist. In the lab, we run a whole panel of tests just on this sample's integrity. We test for oxidants and nitrites, we check the pH. If it's super acidic or super alkaline, you know something's been added. But pay close attention to the last two. Creatinine and specific gravity are your best friends for spotting dilution. If creatinine is below 2 or specific gravity is basically that of water, you know the sample is invalid. It's just too dilute to give a meaningful result. This brings us to our final, and you could argue most important section, Part 4, Analysis and Reporting. This is where everything you've done has a direct and often profound impact on someone's life, their family, and their career.
I want you to really let this sink in. Every single one of us in this field has to remember that the results we produce have massive psychological, social, economic, and legal consequences. Our work is never just about numbers on a report. It's about people. The weight of that responsibility is enormous. So, let's talk about the results themselves. The single most crucial distinction you have to understand is the one between a screening test and a confirmation test. A screening test is just that, a screen. It's fast and sensitive, designed to catch the possible presence of a drug. But, and I cannot stress this enough, you never report a positive result based on a screen alone. Ever. It must be confirmed using a second, more specific method, and the gold standard for that is gas chromatography/mass spectrometry, or GCMS.
Now, this table just gives you a little peek at what we're looking for. I won't read the whole thing, but I want to point out a couple of key things. First, look at that insane detection window for cannabinoids, up to 90 days for chronic users. But even more important, check out that last column, common false positive source. For opiates, something as harmless as poppy seeds can make a screening test pop positive. And that leads us to a really important clinical update. And this is just a perfect example of how science has to adapt to the real world. Think about it. Someone eats a poppy seed bagel for breakfast and could get a positive opiate screen at the old 300 ng per milliliter cutoff level. To prevent this exact kind of false positive, many labs have now raised their screening cutoff for opiates all the way up to 2,000 ng per milliliter. It's a smart, practical change that protects innocent people from a potentially devastating false accusation. So let's bring it all home. When you think about all the legal issues, the personal stakes, the careers on the line, what is the single most important principle in all of drug testing? It's the absolute, unwavering integrity of the entire process. And that integrity is built, brick by brick, on the foundation we've focused so much on today, a perfect, unbroken chain of custody. That is what makes your final report stand up, not just scientifically, but in a court of law.